good morning or good afternoon. It is a great pleasure for me to start again with the seminars, with the polar seminars Andrés Barbosa here in the museum. And it's also a pleasure to me to present today, to introduce today Luis Pertierra, a researcher that in spite of his youth, he has a very important trajectory in polar research. And also, it's special, or it's nice for me because now he has just incorporated with us in the museum. He had his master's degree in Sweden with co-supervised with Andrés Barbosa and Anna Justel about penguins. And later he made his doctorate in the UAM, supervised by Javier Benayas. Later, and in this case, he was about environmental impacts in Antarctica. He has been as a postdoc in Chile, in South Africa, and I don't know, it's in any place more? Australia, so in different places. And also, it's important in this context that he has participated in 10 Antarctic campaigns and 2 more in subantarctic Iceland. So now is your moment, Luis.
Ahora. Okay. Thank you, Asun, for the nice introduction, and thanks everybody for coming here. Today's talk is going to be kind of like a sandwich, a bit of science, then a bit of broader outreach, and then a bit more of science. It's going to be about the Antarctic tree of life in the sense of all the biodiversity in Antarctica, but not only the diversity itself, but the scientific knowledge around it. It's been a journey that has been part of my life for around 17 years now, quite some time. And hope you like it. So I want to start with thinking about the big questions for humanity, right? The 4 things we need to think about are what we know, that we know, that what we know that we don't know, what we don't know that we know, and what we don't know that we don't know. Okay? Right? The known knowns, known unknowns, unknown knowns, unknown unknowns. Okay? So this is critical for identifying all the knowledge and the knowledge gaps in the system. So for Antarctica, in order to approach these questions, a few years ago, partnered up with another researcher, among many other people, of course, but in particular, just highlighting here Joaquin from the museum, who has been exploring these questions broadly for many groups and many systems in the world, and we wanted to bring it to Antarctica. So a way to approach this question around what we know and we don't know, this framework offers a system to think about compartmentalize biodiversity or ecological science in different dimensions, right? So these dimensions are species diversity by itself, right? It's called Linnean, after the father of cladistics. And the second one, Prestonian, regarding populations and community population dynamics. Wallacean, which has to do with distributions. Darwinian, evolution and the evolution of characters. Ronkerian, the evolution of functional traits, the roles. Hutchinsonian, which is the expression of the characters in response to abiotic stressors, and Eltonian regarding the biotic interaction. So by these categories, I really hope that all of you regardless of what you are researching, you feel identified with some of these or probably all of them, right? But this is just a way to compartmentalize what to do. And with these categories, we can identify all the knowledge, the flows of information, all the data available, missing data, and reanalyze that. So, but let me first introduce our case study. I'm sure you know Antarctica and the general view of the system. You know that it's a frozen continent, but not so entirely. About 0.1% of it is ice-free. And even in the ice, there's bacteria, there's green algae, and there's different groups that can live in the ice. There's other groups that live beneath the ice, and even some live in the air. So Antarctica can be summarized as this panel, which was done by a big friend of mine, which is a graphic designer with a nice touch. And you can see all the components here, the vegetation, the flora, the microorganisms, and the microinvertebrates. I want to emphasize this picture again because in order to identify the knowledge gaps, we need two things. First, the different disciplines in ecology, and then the different groups, right? So in the complexity of the biodiversity kingdoms and dimensions, we took an approach of simplifying by fauna, flora, and microorganisms. Hope you are okay with that, even though we know that they convey different paraphyletic groups. First of all, in order to study all this diversity, we have all the groups, but we need all the case studies, right? So we needed an inventory of species of Antarctica. And there were previous inventories. The problem is that they were non-centralized. There was inventories for mosses, for lichens, for different groups, and not systematically put together, and some groups were missing. In this case, we combine everything, and for many groups, this is the first account, a checklist of diversity, right? So in order to fill the gaps, we start by filling the gap of diversity inventories. This is important because when you start doing conservation or research actions, it matters a lot, right? Representativeness. As with humans, different groups would like to be represented and not neglected, right? It's important for area protection, right? Protected areas. For instance, in Antarctica, the first protected areas were protected after penguins and mosses. Many other groups were neglected. And by knowing all the organisms there, we can finally include them. There's many researchers involved here. I just put a photo of a few ones who have been helping, and they are the true taxonomists. They know the people who know about the groups. This is the look of the tree. It's not a phylogenetic tree. It's just a combination, an amalgam of trees and put together to look at them. And by this, we can start seeing some gaps. And it's interesting to see which groups are omitted from Antarctica. And in fairness, there are not that many groups that are missing in Antarctica. Maybe a fifth phyla of animals. Plants are unbalanced, and we'll see. For most groups, everything is there, just with a few representatives, maybe certain lineages with a specialization to cold systems, but it's a good number. And interestingly, there was also no inventory of microorganisms, and we talked that inclusiveness, right? So if you start doing an analysis of knowledge gaps and you start omitting a group, then you're doing things wrong, right? The problem with microorganisms is that they're not that easy to cover, right? In eukaryotes, we're talking about 2,000 species, which for a whole continent is not that much, but it's a reasonable number. Still, we went to analyze all the ASVs, the amplicon sequences for microorganisms, and we cover all the diversity that is known to Antarctica, and we will see that, right? So these are all of our study cases. Right, now that we have the guest stars of the show, we can look at start looking at the stats, right? And you can see for the species accumulation curves that there's different paucity levels for groups, but you could say that in none of them there's been clear saturation. Most of the findings come after the late 50s in 20th century because of the Antarctic polar year and the first recognition of Antarctica as a continent for peace and science, and a lot of research over the 60s and 70s, and there's more groups to be found in all diversity groups. And more than that, it's also interesting to look at the spatial inventories and how all the areas of Antarctica have been surveyed. All what you see here is ice-free areas. So you can see that there's not much of ice-free areas in the continent, right? But they are quite widespread. It's just the small areas. For instance, Leo studies lichens in nunataks, in remote areas in the peak of mountains where you'll expect nothing to live. There's a few organisms there. Then there's also the coastal areas where it's much easier to have biodiversity because of the ameliorated conditions. But you see the coverage. So the more red, the less certainty of the spatial inventories. And in some areas, these gray areas of the transantarctic mountains or Ellsworth Land, there's simply no surveys because there's no records there. It's extremely difficult to find. And another issue is mobilization. There may be records, but they are not accessible to science, to the final user. So by not only doing additional surveys, but also looking at gray literature and developing artificial intelligence systems to look at all reports, we could cover the system. Non-surprisingly, the area of the peninsula where most of the Antarctic stations are placed is where you find the cream areas with more completeness. It's a huge agglomeration of scientific stations, and in the Ross Sea, where the American station, Italian, New Zealand have research, these are the best known areas. And for some groups, like of microorganisms, bacteria, and viruses, there's simply no spatial inventories. There's just a few environmental DNA analysis. So moving on into a bit more aspect of outreach of Antarctica. So let's look at all the components. We have fauna, about 400 species, from which 20 species are vertebrates, higher vertebrates. You know penguins and seals and breeding seabirds. They represent 26 species. So it's about, let me do the math, 10% of the biodiversity, of the animal biodiversity in the continent. This is important to know. They have an important ecological role, and they are quite interesting, but they also receive a lot of research, and they're very well known, and they kind of act as umbrella species. And there's a lot of tiny things, crypto-organisms that live in Antarctica. For instance, there's almost 200 arthropods. And this is the group I typically work with Colembola, hence why I have so many photos of this and not borrowed from internet. There's a lot of mites, which we haven't identified ourselves because we don't have the capacity. Maybe through artificial intelligence in the future, we will be able to solve the problem. And there's a few dipterans as well, just 2 natives, and only 1 flies, and then 2 aliens, 1 that flies and 1 that doesn't. A few crustaceans in freshwater systems, but not that much. Still, 200, good number. Then there's, so these were the mesoinvertebrates. Even if you look into more detail, it's tinier things and higher diversity when looking at the microinvertebrates. So only considering rotifers, there's 70 species in freshwater streams, lakes, and 70 tardigrades that we know of. These are a few examples of the places you can find them in this areas where the snow melts in Antarctica. I want to mention a group that may be overlooked, which is ecto and gastroparasites of vertebrates. So going back to vertebrates, they are a few species, but they are also an ecosystem on themselves, right? Because they host other organisms. And I want to remember Andres. He was involved in this research, and he helped with the identification of penguins. We all know the penguin species there. But more importantly, he also studied the ecology and the biology of the penguins and all the gastroparasites. So much of the research on the things that live as parasites of others are thanks to his work. And in summary, we have 81 nematodes. I just combined the number here because there's also a good number of nematodes living freely, but some also parasites. And then plathelminths and acanthocephalus, which are purely parasites, and also ticks and others. Maybe Josabel or others can tell you more about this. And another group covered here was the black box of SAR protists, Stramenophila Alveolata and Rhizaria, and then other unrelated groups like amoebas. And for these, they were not listed at all, so we look all the records, and we got around 300 diatoms, around 130 protozoans, and not many other groups. There were a few amoebas, just one, maybe two, and not other groups. For plants, another charismatic group. There's only 2 native plants, and this is a grass and a pellworth, so a monocot and dicot, and then a few aliens. So I've been working with the aliens. There was one that was eradicated. We eradicated a few years ago. Then there's the issue of Poa annua, which is also still there, right? The Polish National Research Program are still dealing with that. But in contrast to higher plants, where are only 2, very poorly diverse. In the continent, bryophytes, bloom. There's a lot of micro-habitat conditions that favor their establishment, and there's 130 species. Forgot to put the number for the liverworts and hornworts, but there's around 40 of them. And they're very interesting. Some of them are highly cosmopolitan. You find them in many places, like Marcantia, and they live in freshwater streams where the snowmelt happens. They are also associated with geothermal conditions. There's one moss that is not only endemic to Antarctica, but also endemic to one island that Spain has researched, Deception Island. It's a volcano. And there's good number of diversity. Green algae, another group that doesn't have a consolidated checklist, but this is the number of species we found through looking at the literature, about 150 species. I include cyanobacteria here, even though through ASB recognition they also yield results, but I put them together here because typically in ecological studies they are studied together. And then the true winners of colonizing the Antarctic continent among the eukaryotes, the fungi, around 1,000 species known to science at the moment. Half of them as lichenic or lichen-forming, and half of them are non-lichenic. Probably considering the use of new array techniques, the number of soil fungus are going to grow exponentially with additional research. Yeah, and interestingly, there's a few mushroom-forming species. They're quite cryptic. I've barely seen once or twice, but there's around 50 species of them that happen to produce a mushroom. And they live in different soils, and and different environments. So there's the ones that dominate the drier conditions and soil-forming conditions of the maritime Antarctica. And here some researchers can tell you more about that. There's different groups present in these environments, but they can also be present in one of the most extreme conditions on Earth, which are the dry valleys, where they can endure very harsh and dry conditions, like this example by Leo. And even they can even live under the rocks in some complex symbiotic associations. And of course, the work of Asun. Some photos here provided by Kevin Hughes of endolichic microorganisms and fungi living in Antarctic environments. And to finish with this section, the number of prokaryotes, which we still don't know. Our next goal is to, with all the ESV we collated, and looking at additional sources, thinking if we can do the same graph showing the species accumulation curve, but having a similar estimation of how much diversity of prokaryotes Antarctica hosts, right? But at the moment, through all the combination in Silva dataset, there were identifiable a number of about 2,000 identifiable bacteria genera. So it's the same number of genera for bacteria than species for eukaryotes. So tenfold, not tenfold, one hierarchical level fold of diversity for bacteria, and then a bit less of archaea. And the very last thing, viruses. This is the Wild West. Not much done. Some of the work done by Spanish groups from SIC and WAM, where they found high diversity in Antarctica of viruses, and but still a lot to know. There's a lot of viruses and funguses in all systems. For some reason, they diversify a lot, and also the ice has preserved very old viruses for thousands of years that they can revive, right? So they called zombie viruses, and there was been also discussions and arguments about researching them or not, because people are concerned that if you melt viruses in the ice, they could start a new pandemic. But well, there's been some research on that. And this year was hectic in Antarctica because highly pathogenic avian influenza arrived to Antarctica, and there were mass mortality events for seabirds in Antarctica. So Antarctica is not an influenza-free area. There's different groups working on that, including Spanish programs. Right. So this is the diversity. I just wanted to present that because I find it fascinating. But now we can look at the knowledge gap, information knowledge gap filling estimates. So, okay, we have all this diversity. How do we look how much we know about this diversity? Well, we went through a dual system of indicators and expert elicitation. Firstly, through indicators, we went to the data banks of ecological knowledge for the different ecological disciplines. You could argue that you could use a different one, but we chose one per section. So for instance, for spatial occurrences, we got everything that's in GBIF, even though there's other atlases. For genetic information, we went for GenBank, even though, for instance, for Colembola, probably it's better to use BOLD, but we use that. Then for traits, we use any repository available, and for several groups, there's no repositories at all that we know of, centralized, but there were a few that were. And so for plants, we use TRI, which has entries for vascular plants, mosses, lichens. What else? We use Globi. I recommend for you to have a look at Globi for biotic interactions. It's just a website that instead of giving you an occurrence, it gives you an occurrence of an interaction, right? So we downloaded all the interactions south of 60 degrees and got all the individual entities. So all these nice looking but hard to read graphs show the composite of ecological knowledge at the species level for all the eukaryotes in Antarctica. So ideally, you want to be here in the center, right? Because it means that for that organism, we have some genetic sequences, some spatial records, some measurement of tolerances, some functional traits. So you could guess that that species is probably going to be a penguin or a seal, but it represents a minority. It still is relevant because once you know these ones, you can start thinking about using them as model organisms. And also, you can look at the disparity between graphs. So for instance, some groups are more studied in terms of spatial distributions, and others more in terms of tolerances. And so the information cannot be integrated easily, right? Different groups have different graphs. Naturally, you see less and less for more cryptic groups, and these shapes give you some information. In the lower part is the same information, but standardized by different units. So for instance, rather than seeing if a species have genetic sequences in gene bank, let's look at how many, right? So the average of genetic sequences in gene bank for each organism in the continent, and you see the variation. Same for the different other charts. And the fraction, right? So having a high number like this one, like megafauna, all the species have DNA sequences. So all the vertebrates have some DNA sequences. And for other groups like green algae, there's not so much. And so we know where the knowledge gaps occur. Right. The problem is that indicators are just indicators. So you will raise your hand and say, why you chose that database? Why you went through that indicator? Yeah, that's right. It's just a proxy, right? It's not ideal. It gives you an idea, but you could question that. So we combine this through with an expert elicitation, in where we went through the experts and asked them to meet together and discuss the knowledge gaps from the different perspectives. So we put around 25 people that cover different expertises, both in taxonomical groups, but also disciplines, and ask them to relativize their knowledge and rank them. So here's the summary information. This is at the group level. Non-surprisingly, there's more knowledge in vertebrates than any other group. But the interesting part is the subtle differences between groups, right? So for instance, Hutchinsonian, which relates to abiotic tolerances, has slightly lower values than others, maybe non-significantly, but there's probably more caveats there. And one of the reasons is that now burning a penguin is considered unethical to see how long it takes to die. And I'm not advocating for that. I'm just explaining that different research actions cannot be applied with the same impunity or the same freedom for different groups, right? So for invertebrates, we see that we have a good coverage, and in contrast, we have a good notion of abiotic tolerances and evolutionary relationships. For embryophytes or plants in the higher plants and mosses, we have a very good coverage of spatial distributions. Fungi with TK, and especially in terms of species interactions, even though in the case of lichens, we do obtain that. And these graphs show color, but I don't know if you see that the scale also changes, which matters a lot, right? So we go from 0 to 100% to the estimate of viruses 0 to 4%, right? So the general consensus with the experts is that we know barely nothing about viruses in Antarctica, almost nothing in prokaryotes, but we know a little bit more about functional traits because they are studied at the community level, so we can identify functions even if it's at the community level. So this gives you a snapshot of the knowledge in the continent, right? So apart from collecting the indicators, or just because we were collecting the indicators, we thought like, oh, we got the indicators, maybe we should explore them further, right? So here in a collaboration with Chris Fraser of Otago University in New Zealand, we got all the genetic sequences in the continent and started looking at richness patterns. But now richness patterns not in terms of species, but rather in terms of genetic diversity, right? And we see some patterns in the continent which have to do with the extremicity of conditions, but also influenced by survey effort, and they can be used to test some hypotheses. So Creed is a big advocate that Antarctic biodiversity survived through glacier periods in geothermal refugia, and by looking at these sequences, she can test that. She published last week in Antarctic Science a paper showing that she found more genetic diversity around volcanoes. So seems to be confirming her ideas. So these are the kind of studies you can attain with this. Working with Chilean colleagues, this was my previous postdoc, when you have the Wallasian information or the spatial distributions, and you also have the genetic information at the good level using molecular techniques that I barely understand, we got all the necessary information for modeling species distributions in the future, but not only accounting the current environmental envelope, but also the genetic plasticity levels. And this is unpublished, but Lucila is working on the skuas of Antarctica, which have had a long debate around how many species they are. Ornithologists or twitchers are crazy about finding, like, oh, spotted this particular subspecies and then put it in the checklist on the book. They may be unhappy with that, with the findings of this study, that we find some subspecies not to be considered discernible. And also there's a very interestingly high degree of hybridization among skuas in Antarctica. So for doing the species distribution models, we accounted all of that, particularly when thinking about the resilience of species withstanding climate change scenarios. And importantly, how the different lineages are going to fare, right? And naturally, some lineages that are more adapted to warmer conditions are going to outcompete the ones that are less so. Abiotic conditions don't have strictly, in the strict sense, global repositories, and this is a huge knowledge gap, in my opinion, in the context of climate change. The very notion that there's no website where you can see the tolerances of organisms that have been measured, that I know of, and especially for terrestrial organisms. Maybe you know about marine organisms that have repositories. Anyways, there's no centralized information, but there's some publications that have done reviews. So we did a review for all the colembula in the world, and because we are Antarctic biased, we related this with the colonization to the poles. So we did some ancestral niche reconstruction using a mix of techniques of molecular and spatial modeling in order to figure out the ancestry of colembula. I don't want to, I don't aim to talk about this work. I was involved and was interesting. I just want to showcase the kind of studies you can do if you have these kind of repositories, right? Multi-species data that allows you to do multi-species analysis where you establish patterns and processes. And as a curiosity, interestingly, in Collembola, we found that Collembola species that are related to polar ecosystems have lower thermal tolerances, which is kind of obvious. But interestingly, they also have lower maximum thermal tolerances. And we thought this could not be the case because there's a high conservationism in thermal tolerances, but it seems that there's a trade-off in the machinery for dealing with, I guess, the heat of proteins and so on. They adapted to cold conditions and they had to lose some genetic weight somewhere. Well, this is what we advocate, and so we find this. Another example of cases where you could benefit from having robust repositories of ecological information. Now going into the case of functional traits, it's in the study of invasive species. My research in Antarctica has dealt with invasive species from all the angles possible. So with Collembola, I'm working with Collembola because I'm interested in the invasive species. With plants, same thing, and so on. So one question we wanted to study a few years ago was the patterns of invasion in polar systems from the perspective of functional traits, right? So whether alien species in the poles share the same functional strategies. You could also test natives versus aliens, so there's many different things. But in this case, we were just simply interested about the same properties they share. The problematic is that retrieving the information took forever, and there were no repositories, but eventually we managed to get data for 400 vascular plants and starting to do some phylogenetic path analysis where we identified as a response the number of invasions that could be the explanatory factors of drivers. Some obvious, the species that invade the poles have to have cold tolerance, lower cold tolerance than average, but also some functional traits like specifically leaf area or plant height, they tend to be smaller to colonize the poles, which is interesting because it goes against the Bergmann's rule, but in plants it's like that. And so another example of that the better environmental data set you have, the more questions you can answer and reanalyze data. Lastly, another example. With biotic interactions, we don't know that much. Again, going back to biologic invasions, which is my thrive, we look at the alien species in Antarctica, Poa annua, the alien vascular plant, and we were wondering why this species is so successful, and did analysis through all over the world with the endophytes and rhizophytes association. And what we found is that the highly invasive potential of Poa annua to establish in very different conditions. So this plant is in Antarctica, but I have also seen it in the Australian outback in the desert. So how come this species can be in Antarctica and in the desert? Well, it happens that it's very plastic in the association with endophytes. So we argue that that could be the case. But again, it's just a particular case. What I want to emphasize is that by having good inventories of biotic interactions, you can start establishing patterns and processes. Getting now to the conclusion, why doing this research? Well, I want to emphasize that even though there's far fewer species in Antarctica, that doesn't mean they don't bring any, they don't bring ecosystem services to humankind. And we did a review of ecosystem services provided by Antarctica or by Antarctic ecosystems, and nearly all were also present. Apart from forestry and agriculture, every single ecosystem service was present in Antarctica, and some of them, like regulatory, like maintaining the air masses homeostasis and so on, were extremely relevant. This is important because Antarctica and places where there's no human populations, they tend to be overlooked or neglected in analysis like IPCC or IPBES reports, although it's changing now, but it has to do that things that happen in remote places matter a lot. So this is just a visualization of the ecosystem services and the investment of research done over the years in Antarctica. This is the same figure as before, but providing a summary, all the different aspects and assessment where we synthesize what we know and we don't know. So for instance, in vegetation, species spatial distribution and competitive, well, not only competitive, biotic interactions are highly studied, but in contrast, abiotic tolerance and phylogenetics are not so well known, and they have good repositories, which is not the case to other groups. And vertebrates, we have a good coverage of everything. There's still more to know, but it's not as bad as other groups. In microorganisms, there's a huge upstream of ecological studies at the moment in Antarctica, but a big knowledge gap is the metabolomics of these microorganisms in Antarctica. And microinvertebrates, we face that they are ideal model groups for the study of abiotic tolerances because they are easy to study in the lab, but there's very, very, very poor knowledge of population dynamics. We know if the penguins are decaying or not, or the vegetation is becoming greener or not, but we have no idea what's happening with the microinvertebrates because there's no monitoring station, although it's changing a little bit. So conservation in Antarctica is a huge service to humankind, and we advocate for that. We've been very insistent in policy forums and journals, and just to show the applications of this meta-analysis of knowledge gaps is that you can identify policies, right? For instance, all the groups that we discuss here, they have these different scores, and then you can identify, okay, I'm interested in climate action. Then which groups should reinforce research and which ecological disciplines are more demanding, right? This is just an example, so these are different examples. Maybe, for instance, for bioprospecting is another question that if we don't have repositories of functional traits or functional knowledge, then bioprospecting is hampered because we cannot identify bioproducts or aspects of biochemistry of the biodiversity that could benefit the humanity. And very briefly, just to say that Spain is very present in Antarctica, that has been doing research for quite some time. This CSIC, I don't know how to say CSIC in English, is also present with Polar CSIC, and yeah, it's been growing with some variations over the years through economic crises. We are doing some efficient research, which is good and bad at the same time, because by having struggles with funding, we're becoming very creative in producing high-quality science, but at the same time forces you to research on topics that don't demand field cost or experimental cost, right? So we end up doing a lot of modeling and reanalyzing, but there's other research that require field studies or lab studies and cannot be done with funding. Just to show that we tend to be associated with countries where we have cultural affinities. We also have a association with countries that are closely close to Spain and in the same, well, the European Union, and other countries that simply have a lot of investment interest. Here's the snapshot of the biological research in Antarctica with identification of the different groups. Here, there's Andres' research in penguins or seabirds, our works in soil invertebrates, Leo and probably Asun is also there in, I'm going to say flora as a whole, and other groups. Maybe even Sergi is somewhere there with, probably, yeah, with Conchita studies, but this is the different groups. In this case of the research done by Spain, not covering all the dimensions of research in Antarctica is not necessarily a bad thing, because we are in a global context and other research programs do a lot of research in other questions. So it doesn't have to be, everything doesn't have to be filled. Doesn't matter which country does. So this is all that's today. Some conclusions. The value of consolidating ecological data, centralization, mobilization, and reanalysis. There's opportunities for cryptic groups that are less charismatic. For charismatic groups, it's important to identify which questions are either the most, most compelling and most pressuring, and identifying all these knowledge advances is helpful with limited resources to identify species that can serve as model groups, no? And the same for priority areas. Spain, it's to be continue in integrating in the international arena, trying to minimize redundancy, so doing complementary science and not redundant science, and maybe keeping the leadership in areas where we are strong, for instance, in the South Shetland Islands where the stations are, or the geothermal sites, because there's few volcanoes and we happen to have a station wisely or unwisely placed on atop of a volcano, and glacier systems, as we know. Thank you.
So thanks, Luis, for your interesting and also nice presentation. And now we have time for questions.
Okay, thank you very much, Luis, for this overview. It's really very useful for all of us. I wanted to ask you, or to make a reflection about the level of knowledge in the Darwinian area, in the evolutionary area. I wanted to put on the table also the importance of the USA, the unidades de significación evolutiva, the significative, bueno, dilo tú en English. Those populations that maybe are already in another evolutionary route. So we don't have to forget that one species has a lot of populations, and when we are working in this kind of data, we need to talk about the species. But yeah, we have to make the zoom because some populations are already with local adaptations and so on. Do you think it will be possible to introduce that in your data, at least for the macro animals? I don't know.
Well, yeah. For introducing in the data set, maybe looking at genetic sequences instead of species could be the way. Just to say that I didn't talk about that, I fully agree. I published a few years ago a paper about the cryptic speciation in gento penguins, where we found that there was genetic differences, and we are advocating to splitting one species into 4 species. First explorers already talk about 3 species, But then we thought like, they were miscommunicating to one to another. There's probably just one because it looks the same. But probably they had a very good eye, and they already named them. So we wanted to revert back to that status. Anyways, what I'm trying to say is that by looking at the species level, you may overlook and end up making mistakes from knowledge gaps. So I fully agree with that. I didn't talk about that, but yes. So there's going to be changes at all levels. Although how complex will be to work at that unit, it's going to be rough.
It's difficult.
But I also wanted to emphasize that even very well-known groups, this is relative, right? We know a lot about penguins in comparison to viruses, but it doesn't mean we know everything about penguins, right? And you can always also consider the best-known groups, but still the genetic of seabirds is quite basic at the moment, right? We're starting to look at the skuas at the moment, and so on. But completely agree.
You are doing very well. Maybe just an idea, maybe for some species to give a list of populations and which populations are in very different conditions. Just to know, because this is already saying something. Yeah.
Okay, I have to say that the seminar is followed also by YouTube, and so we have some questions from there. So first, one of the first slides was a phylogeny. I wonder what it would look like if the biomass of the main taxonomic groups were taken into account.
Yeah, that will be. In fact, we thought about at some point to these graphs that are histograms. We thought about plotting them using the heat map of the tree, but we felt that it will lead to misinterpretations, and so there's different ways to mind when plotting this, because you have to account the ecological role, right? There's few species there, but they have an important biomass, and they also represent the apex predators, so they modulate the community from atop, and so yeah, it's a thing to mind. Just to say that the approach we took here is that let's use all the species as the unit of analysis and give them the same treatment. I'm just not saying that we are all the same and should be treated the same, right? There's a lot of aspects to consider.
Okay, and we have another question. The dry biomass per day need to be shown because penguins, for example, are only around for a few months, just to get an idea, because most of the biomass cannot be calculated. That's another question. I don't know if it has to do with that, but why we included seabirds is because they spend partly time in land, right? So we could have said that seabirds, penguins, and seals are not Antarctic diversity. They are marine things. But they spend quite some months there, and they bring nutrients from the sea to land. So we include them. It's just that it was quite tricky to split, to set the limit between marine things and terrestrial things. So now we have more time for questions in the audience. I have a question. So if you can choose, which strategy do you consider that is better? To look for more the diversity, to try to describe all the diversity, or to describe better the groups that we know actually?
Well, a thing that's been discussed is that if you're doing research in remote areas, right, and if you are specialized in one group and are going to do all the logistics to get to one area, like the top of the mountain, and you are specialized in a group like tardigrades, there's been conversations in the Antarctic policy arena that maybe bring all the experts so that you maximize the benefits of the visit. So there's more and more an interest to do multidisciplinary teams and not everybody studying things on their own, because it leads to higher effort. It's obvious that we tend to be focusing on what we're interested about, but that's one thing to mind. And then regarding your question, well, what we advocated in a recent discussion is a mixed formula where model organisms are chosen for, especially ideally if you can identify model organisms that represent functional groups, so they could be used as biosentinels, and you could put more and more research on them and have a disproportional effort on them, so that you maximize with the minimum resources the knowledge you get for the functional group, but at the same time combine that with a baseline strategy of filling the gaps for all of them, because if you focus too much on model organisms, you may see that you fall into missteps. For instance, what we say, like you may focus on one species, like let's say Adelie penguins, which don't have any genetic differentiation. There's a very good transfer of genetic material between all the populations. So regardless of the placement in Antarctica, they have gene flow. But then you have others that have hybridization and others that are completely isolated even from a few valleys distant. So each case is different. So model groups are good in a world of limited resources, but they lead to these issues. So I will say like half.
Okay, thank you.
Bueno, primero gracias Luis por la charla y disculpa mi ignorancia porque yo no sé de biología. Entonces te quería preguntar si este conteo que habéis hecho de todos los, esta revisión de todos los estudios que se llevan a cabo sobre especies en la Antártida, ¿incluyen también el ambiente marino? No, es solo en tierra.
Sí, vale. Es una cosa que lo pensamos de partida y los editores, revisores, lo primero que nos dicen, ¿por qué no has hecho el medio marino? Yo, pues es que son un montón de bichos. Son muchos ecosistemas, muchos sistemas. Entonces, si quieres abarcar mucho, poco aprietas, ¿no? Y empiezas a cometer ya errores. O sea, yo este trabajo creo que está bien, pero ya a estas alturas lo arreglaría de nuevo porque aplicaría otros métodos y otros más de detalle para ir corrigiendo todas estas cuestiones que van saliendo sobre las marchas. Entonces, es verdad que no hemos abordado el medio marino, pero una cosa que sí hemos intentado es hacer como un framework, o sea, un sistema de referencia para la evaluación sistémica de vacíos de conocimiento que pueda ser aplicado a otros sistemas. Y de hecho, no eres la primera que me lo comenta. Bueno, igual lo siguiente a donde va esto es al medio marino antártico.
Vale, es que como he visto a Conchita, y sé que Conchita Ávila, ¿no? en la red de contactos que se establecen a nivel nacional, ¿no? Y sé que ella, bueno, los conchitos van allí a bucear y a estudiar el ventos marino y las comunidades que hay allí. Pues pensaba que quizás sí que estaban incluidos. Más que nada también porque desde el punto de vista físico, ¿no? Nosotros estudiamos los sistemas glaciares y también, bueno, pues con el deshielo que se está produciendo en algunas regiones, pues me interesaba saber cómo el exceso de agua dulce que se aporta por el incremento de este deshielo podría estar afectando pues a este tipo de comunidades o a la distribución de las especies, bueno, y eso, pero bueno, ya me espero entonces al siguiente step.
Bueno, quien tome el liderazgo, quiero decir.
Bueno, yo te usaré a ti como vector para acceder a
pero
eso.
Pero sí que es verdad que también lo hemos discutido mucho, que no son sistemas independientes los terrestres, o sea, dependen mucho del medio marino. Muchos de los recursos, los nutrientes vienen del medio marino, luego hay nutrientes que van al medio marino. Entonces, muchos estudios mezclan medio marino y terrestre. Y hay organismos que encima están en los dos sistemas, con lo cual, pero fue un punto donde acotamos y decidimos parar.
Thanks, Luis, for this nice summary. Now that thinking about 6 or 7 years that this work has taken, just for the terrestrial, imagine with the marine. And I mean, I think the values is the value of this kind of studies is what you said, is identifying some gaps so we can fill them. So imagine that now you are in the position of deciding to invest some money from Spain. And so I really like your graphs or summarizing of what Spain is doing and this strategy of filling in some, and perhaps we'll put in the windows in the building or something like that, or roofing some parts. So what could be a good priority for Spain to invest in Antarctic research right now? 1 or 2 things that need research and for which we have capacities.
That's a fair point. I guess I will go to this graph, right? So this is the priority knowledge gains overall, right? So the first thing is identify what's the Spain priority or Spain strategy, where we are interested most. Maybe we are into climate action because we are in a country that's going to struggle with that, or some other aspect of biodiversity. So first define which area is important to Spain, and then I will combine it to this graph, right? There's another one with terminology made by Felipe about the main hotkeys, the topics that have been researched in Antarctica, and then combine them because that will show what we are strong, right? The things that we have long trajectories of research groups studying, and then combine with the other graph and say like, okay, this is the knowledge gap. This is where we are strong. Maybe we decide to take a leading role in filling that knowledge gap with these research groups that are strong about, right? So, and then if you want specifics, I'll say that Spain has done quite pioneer work in ecophysiology in Antarctica, both for flora and fauna. And what else? I tend to think on my own contribution, but there's also, for biotic interactions, I'll say there's also quite pioneer outstanding work with penguin, I'll say penguin, but seabird researchers. I'll say that biotic interactions, we do have an important contribution.
So you will keep doing as we do?
Hmm?
You will keep doing as we do, what we have been good at before. Que no te mojar, vamos. Thanks, Luis, gracias.
But you could do it simply by overlapping this graph with the other graph, and then we were thinking in the international context, but that's a good idea to figure out. Oh, there it is.
I just would like to add a comment that there are also new scientists from other countries that come to Spain that can add more information to those graphs.
Yes.
So please don't forget the early career.
No, yeah. This is just a visualization. This is bibliometrics, and what Felipe did here was to limit. So it's only showing people with more than 10 publications. So there's a lot more of research, and 10 is not a magical number where you qualify as suitable to be seen in the graph. It's just to highlight. I don't think it's that relevant to that graph itself.
Yeah, just in case.
No, I think the hotkeys, the topics, the keywords are more important. No? Where are the keywords that Spain? I should have put that graph. The hotkeys where Spain has been producing research and how they relate one to another, and the clusters, no? And so we can define the disciplines where we are strong.
So, more questions? No? So thanks, Luis, and thanks to all of you for coming here. And only I would like to remind that we have a new seminar of Polar of 28th of June by Eva de Andrés that is there, and it's about polar ice or Arctic, no? In Arctic. So we will wait you. Okay, thank you.